So yesterday, we heard some great presentations on using synthetic biology to accelerate the study and vaccine development for SARS and avian flu virus. And today we are going to continue to hear some good presentations on developing new therapies using synthetic biology. And our first speaker is Professor Wendell Lim from UCSF.
Okay, well, let me start out by thanking the organizers and the people here from UST for inviting me to this really wonderful meeting and putting together a really amazing group of people. I also want to thank all of you for making it here this morning, especially those of you who went out to karaoke or dancing last night. Today, what I'm going to talk about is some of the work that we've been doing that's aimed at moving towards sort of a family of biomedical applications of synthetic biology, but really trying to ask whether or not we can learn how to engineer cells that can carry out what I'd call smart tasks that could have some therapeutic value. So in modern medicine, we basically have two families of strategies for treating disease. We can treat things with drugs, or we can treat things with surgery. And both of these are amazing and very effective in a lot of ways. Some of the pros about using drugs to treat disease is they're very easy, non-invasive, non-traumatic. But some of the cons are that you give a drug to someone, it has systemic action, and so you can have a lot of side effects. Surgery, of course, has an opposite sort of set of advantages. It is by nature very specific. You act in a local and surgical fashion. But it's also very traumatic, invasive, and difficult. Of course, what I think a lot of people would like to see is, can we make smarter therapeutic agents? And what do I mean by smart? I mean things that, for example, have the convenience of, say, a drug, but that, for example, can target to very specific sites, that can do some sort of computation, make decisions like a surgeon could about what the situation is and what action should be taken, but then to take this information and decide to either deliver a payload or execute some sort of action in a very intelligent and localized fashion. So, of course, in today's world, this sounds a lot like science fiction, and this is, of course, what the vision of movies like Fantastic Voyages was, to be able to shrink down a bunch of surgeons or doctors and inject them into a body. In today's world, a lot of people are interested in applying nanomaterials. Can we make nanomaterials or nanoparticles that can do things along these lines? What I'd like to talk about today is the fact that we know that there are machines that can do this kind of thing, namely living cells. And the question is, can we actually harness this sort of power? And I want to just show you this movie that some of you may have seen before. It's an old movie from the 50s. But it shows you a human neutrophil among red blood cells. And what this cell is doing is it's able to detect signals from this bacterium and mobilize in a very precise way its actin cytoskeleton so that it moves towards that. And what you'll see is once it gets there, this cell will then execute this action of phagocytosing as well as releasing killing agents that will eliminate that bacteria. So this is exactly the kind of thing that we're talking about. A very smart therapeutic agent, the cells of our immune system, that are able to target specific sites or read specific signals, make decisions about what to do, and then execute very complicated actions, in this case things like mobilizing this coordinated motion as well as phagocytosis. And so one of the questions that we, and I think a lot of people in synthetic biology have, are whether or not we can actually take cells like this and reprogram them in a way that we specify what the sites of targeting are, what decisions are made, and what actions are executed. So I'm actually involved in two different centers and projects that have this sort of goal. One that I won't talk about is the one from the NSF SINBERC, this Engineering Research Center, where there's a major project on trying to engineer therapeutic bacteria Chris Anderson from UC Berkeley is heading up this project, and you can talk to him if you'd like to hear about that. I'm going to focus on some work that we've been doing in another center. This is an NIH roadmap nanomedicine center that involves people at UCSF and UC Berkeley. It's called the Cell Propulsion Lab. I'm the director of this. And this one focuses on the cells that I showed you before, neutrophils, asking to what can we understand the engineering principles of how those cells carry out their complicated actions, and can we move towards the idea of harnessing that for therapeutic value. So, as I said, this is a center that involves about 15 investigators from Berkeley and UCSF. It's a very tight integrated group that meets at least once a month. Our vision is whether or not we can engineer smart therapeutic devices like the neutrophils, but can we reprogram those behaviors so that we're in control of what they do? And we think about it in several different modules. One is we'd like to be able to control what inputs these cells sense. We'd like to be able to control what computations and decisions they make. And then we'd like to be able to control what actions they execute or what payloads they deliver. Now, just along this line of vision, if you could have different modules that you could hook together in different ways, we can imagine that perhaps we could make cells, engineered cells, that could read signatures that are associated with tumors or metastases, find and label them, perhaps even kill them. Could we detect things like localized flare-ups of autoimmune responses and have cells that secrete anti-inflammatory cytokines that locally suppress that? Could we have engineered cells that would give us very controlled or rapid wound healing? Could we engineer stem cells, for example, that would be used for regeneration, but where we could guide them in a specific way to the sites at which that we want to then trigger their regeneration program? So this is just a handful of different examples of what potentially we could do if we had that level of control. So obviously, the question is, one of the big questions is, can it be done? How hard is it to reprogram cells, mammalian or eukaryotic cells of this type? And I'm going to first talk about why I think it's at least physically possible. The first argument is that there's precedent. That is, cells themselves are evolutionarily very plastic. Evolution reprograms cells in amazingly diverse ways. We know that the same set of parts in a cell can be used in diverse ways to give highly diverse sorts of shapes, movements, and behaviors. Beyond that, not only has evolution shaped it within, say, an organism like us, but also pathogens are very effective at interfacing and changing cellular programs for their benefit. So the other thing is that as we understand more and more about systems biology of cells, we're starting to see more and more that cellular control systems are, or appear to be in many cases, quite modular, and that they can be hooked together potentially in different ways. And I think one of the keys and one of the motivations of our work is really to try to help to identify what are the relevant modules in cellular systems and what are the ways that they can be reconnected, both in evolution as well as in engineering. Okay. The other thing I want to say is that cells don't have to be so smart to make a big improvement. We don't necessarily have to, for example, have a cell that detects a tumor and then kills it. You could have a cell, for example, that detects a tumor and then mobilizes the immune system, sends out a signal. Because after all, a lot of cells in a body like ours, they don't do everything. They work as a system. And if we could just have a sort of mediator cell that does that sort of function, it could make a big difference. Okay. So I've outlined why I think that it's at least physically possible, but what are some of the challenges? There are many big challenges. One is that the ability to genetically manipulate eukaryotic cells is extremely primitive. We need better tools to do this. I'm not going to address this problem. It's a big problem in biotechnology as a whole. Another challenge is that we need to expand the types of molecular parts that we have, that we understand, and that we can work with. Ones particularly that go beyond issues of gene expression. For some of the behaviors that we're focusing on, we're very interested in parts that are involved in signaling, receptors, protein kinases and phosphatases, lipid kinases and phosphatases, something that you heard about from our iGEM team, GTPases, and other things of this type. But basically, one of the goals that we'd like to do is to help develop a toolkit of cell biological control parts. Okay, so we're just going to focus right now, in terms of what I'll talk about in our center, on the issue of can we engineer the movement and shape of eukaryotic cells. So, as I said before, one of the amazing things about cells is that they can undergo such diverse types of movements and shape changes. These are two human cells, a neutrophil that can move like we showed. This is an axon growth cone used to make the connections in our brain. What's amazing about these really different behaviors is that the parts that underlie them, the parts of the cytoskeleton and the signaling pathways that interface with them are the same, but they're hooked up in these particular cells in different ways. In general, this is kind of a general outline for how these sorts of pathways are organized. You have receptors that detect signals, signaling pathways that process the information and mediate it to this class of proteins called the Rho GTPases that then interface with this engine, the actin cytoskeleton, that gives you force generation and movement. I'm going to talk about two potential control points in this kind of modular scheme that we've been focusing on. One is on trying to regulate the Rho GTPases, which I'll classify as being essentially the master regulators of a lot of the actin cytoskeleton. And then secondly, trying to control these sorts of processes with novel receptors that could mediate novel inputs. Okay, so let me talk about Rho GTPases and give those of you who don't know about them a little introduction. Tanya Kortemi talked about this a little bit yesterday. But GTPases are these switches that can exist in two forms, an off state that is bound to GDP and an on state that's bound to GTP. When these are on, they can bind various effectors that lead to cytoskeletal changes. The conversion between these states is catalyzed by two enzymes. GEFs, or guanine nucleotide exchange factors, catalyze the activation, whereas GAPs catalyze the deactivation through hydrolysis of the GTP. Now, the Rho GTPases are a family of about 20 different proteins in humans, and there are three basic families in them, and they have distinct outputs. The CDC42 family, when this is activated, leads to filopodia, or these spike-like protrusions. You may have seen them, for example, in the end of that growth cone of the axon. RAC is another one that leads to lamellopodia. These, you can't see it here very well, but a sheet-like kind of spreading of cells, like what you see at the front of a motile neutrophil. And then Rho activates actinomycin contraction. And for example, and different inputs in different cells and different pathways activate GEFs and GAPs that regulate these families of Rho GTPases. So how are these used to sculpt cell movement and shape? You can get coordinated control of the GTPases to yield complex movements. For example, in a motile neutrophil like what I showed you, you have activation of RAC at the front of the cell to give you protrusion, coordinated with contraction by activation of Rho at the back, so you can kind of squeeze the cell and it moves forward, and resulting in directional movement. So essentially, these again are the key master regulators of shape and movement. The layer of proteins that really controls these and mediates the diverse set of inputs that controls different cell responses, I would argue are the GEFs, these activators of the GTPases. Of course, there are many different inputs that regulate movement and shape change, and there are also many different GEFs. There are about 100 Rho GEFs, but really, as I said before, only 3 major types of the GTPases themselves. So this is where the diversification comes in. And moreover, if you look at, for example, pathogens, there are pathogens that make their own GEFs that can interface with the host GTPases. So our notion is that during the course of evolution, a lot of what happens is new GEFs are evolved to mediate new responses. And so our question is, can we mimic the evolution of new input responses that are hooked up to these morphological behaviors by engineering synthetic GEF proteins? So we've spent a lot of time studying GEFs and other signaling proteins, and one of the major things about them is that they are very modular in their construction. They have at their heart a GEF catalytic domain that by itself is active and will activate the GTPase. That's what I consider the output domain. But they also have, and this is what varies in a lot of the proteins, different regulatory domains, usually protein-protein interaction domains, and this is what mediates the different inputs that regulate the particular GEF. Even though these differ in their input domains, there are a few common mechanisms that we've discovered for regulating control. One is that in some cases, these regulatory domains can participate in intramolecular autoinhibitory interactions that lock this in an inactive conformation or occlude the active site, but in a way such that binding of different inputs, the right combination of inputs often, will release that autoinhibition and activate the GEF. Another simple way that you can get activation is through localization. Many of the GTPases are localized to the membrane, and so if there are ligands that target this GEF to the membrane, they can lead to much stronger activation. And of course, these two mechanisms are not mutually exclusive. In many cases, you see both of those things. So Brian Yeh, a student several years ago, decided to ask whether or not we could reprogram one of these proteins. This is a GEF called intersectin that activates CDC42. Actually, it's a protein that Reg Kelly, the director of QB3, used to work on. And it has at its heart this GEF catalytic domain, which is also known as a DHPH module. But it has this whole alphabet soup of other domains that regulate its function, in this case in endocytosis. I won't talk about that, though. But what Brian asked is, could we strip intersectin of all this regulatory machinery and now take some other regulatory machinery and link them together? In this case, we took a protein-protein interaction domain that we had engineered, a PDZ peptide interaction, to be responsive to phosphorylation by a kinase, in this case protein kinase A. If this peptide gets phosphorylated, it will disrupt this interaction. Brian made a few different constructs, just a handful of these, and one of them worked very well. It was by itself, this interaction took place, and the GEF was off. But if you treated with pKa and got phosphorylation, you'd see this great increase in vitro of the activity of that GEF. So in other words, we can now link a novel sort of signaling input, in this case a kinase, to the activation of this Rho GTPase CDC42. He went on to show that this could work in vivo. So these are some fibroblast cells that normally don't respond to have morphological changes in response to pKa. This kinase pKa can be activated by increases in cyclic AMP. But normally, again, there's no connection to CDC42, and you don't get a cytoskeletal response. The idea was that if we microinjected this switch, this novel engineered switch, into this protein, now we would have a novel connection, and that perhaps activation of cyclic AMP levels and pKa would lead to a strong filoproteal response. And what we found, in fact, is that if we injected this and then stimulated with cyclic AMP, we could get a strong induction in these cells of a filoproteal response. So in fact, we could turn these on, and if we changed the GEF catalytic activity to ones that were specific for other GTPases, we could get different responses. For example, a RAC GEF domain would give lamelloproteal activation. Okay. So let me now talk about a different way of trying to control these, particularly one that's focused on trying to get much more spatially and temporally specific activation. One of the ultimate ways to control cellular behavior in a spatial and temporal fashion would be using light, because light, of course, you can control in that way. And so ultimately, as I said, GEFs could be regulated by this localization to the membrane. And you can imagine, if you had a light-controlled interaction domain, maybe you could do this kind of thing. And this is where we started a collaboration with Chris Voigt, one of the organizers of the meeting, and his student Anselm Levskaya. Of course, these guys have a long-standing interest in trying to study and understand light-responsive switch elements. So in this case, we turn to plants, Arabidopsis. Plants like this have a lot of light-responsive systems. One of them is this phytochrome that binds a chromophore. It's a heme-derived chromophore. And what's interesting about this is if you irradiate it with 650 nanometer light, it changes conformation, goes on to this on state, but you can irradiate it with a different wavelength of light and toggle it back off. Okay? What's important about this is that there is a protein binding partner, the PIF, shown in blue here, that will only bind and bind very tightly with nanomolar affinity to the on state and not to the off state. So essentially, this is a light-switchable, reversible protein-protein interaction. So what Anselm developed was a way to get very specific spatial and temporal activation. He actually, on a microscope, a turf microscope, globally illuminates the field with the off wavelength, then locally has a movable light source that has the on wavelength. So what I'm going to show you here is a movie where, in this case, we have the phytochrome tethered to the membrane of the cell, and then we have the PIF peptide bound to YFP, so we can follow that. And so what's shown here is this is the light input that's activating the phytochrome, and it's moving around here. And what I hope you'll see is that in this channel, we're following YFP, and you can see that the YFP localizes specifically where this light is. I want to point out that this is not bleed through in the microscope, because you can take the light off of the cell and you still see that, but you don't see that coming through in this field. So we can get localized movement of the YFP to the membrane in a light-induced manner. So now what happens if we tether to this PIF segment a RAC-GEF? Okay, so in this case, what I'm going to show you is this cell where we're going to stimulate with the activating wavelength here at this X. And what you'll see is that it leads to lamellipodial protrusion. What you'll see at the end of this movie is that you'll see this blooming of the cell just at that one spot. So let me run that for you. So again, hopefully you can see that you get this protrusion specifically at that one site that we are irradiating with the activating light. This is another movie that Anselm made. It's the same situation, but in this case, we're lighting it up here, and he had shown the light at that point, but then turned it off, and he's going to turn it on again. You can see he can pull it out, and he can get the cell to extend specifically where we shine that activating wavelength. Okay. So I just want to point out that we think this is a really amazing tool for using light to sculpt cells. Obviously, it's going to be a powerful tool for cell biology, but we think it has potential for doing things like, could you guide axons in a specific path to make specific connections? Could you control wound healing? Other things of that sort. Okay, the last thing I want to talk about is going back and looking more at the global issue of cell motility in neutrophils and whether we can engineer novel input control of directed cell motility. I'm going to talk about actually trying to engineer and change the receptors that are involved in mediating inputs. So in a neutrophil, like the ones that I showed you earlier, many of the signals they respond in chemotaxis towards many different signals. I showed you movement towards the bacteria. Bacteria shed off different signals, including formomet peptides. There's actually a G protein coupled receptor that's used to detect that signal, and that hooks into this guidance system and force generating system that's central to the behavior of the neutrophil. Now, these cells also respond to diverse other chemokine signals, and there are different chemokine receptors in these cells, also G protein coupled receptors, that also plug into these points. So this seems to be another point of modular reconnection in these cells. So what we asked, and particularly Ben Rao, who's a student who's here and had a poster, is whether or not we could put in new GPCR receptors that could guide these cells. And in this case, we collaborated with Bruce Conklin at the Gladstone and UCSF. He's developed a set of engineered receptors called receptors activated solely by synthetic ligands, or RASLs. These are receptors that will not respond to endogenous host signals, but still respond to a small molecule, in this case, clozapine N oxide. But if you engineer them correctly, they can still, on the inside, interface with the endogenous cellular machinery. So Ben asked whether we could engineer these cells with these types of receptors to reprogram neutrophils to sense a novel chemical signal, in this case, clozapine N oxide. So let me just show you some movies from that. This, again, is work that was done with both Bruce Conklin and Orion Weiner, another colleague of mine at UCSF and part of the Cell Propulsion Lab. So what this is going to show you is a micropipette that is emitting this small molecule CNO, or clozapine N oxide. And these are wild-type cells that are not transfected with the engineered receptor. And what you'll see is that they move around kind of slowly, but they seem to ignore this signal. And we have a red dye in here that's also coming out of the pipette. But over here, these are cells that have now been transfected with the engineered GPCR as well as with GFP. So you can tell the cells that have the receptor by their green color. And you can see that they're attracted to this new signal. We move it, they move over there. And this guy here, really green, barrels over in there. You know, in some ways this is a very simple experiment, but I think it shows that this point between these receptors and this complex program that's contained within the cell is a very flexible point where you can reconnect things in different ways, highlighting that kind of modularity and the fact that we can potentially, there are a lot of different G protein coupled receptors that read in many different signals. Can we hook up different signals that would be signatures for various disease states or other things that could link to these cells? We're interested in putting these sorts of receptors into stem cells to possibly use CNO as a guidance molecule to pull them to the right place so that before they differentiate. So some of our current goals are to ask whether or not these engineered neutrophils could find a target in an organism. Can we put beads that are emitting CNO into a mouse and get the cells to traverse the complex environment of a living organism and get to those sites? Another really interesting thing about these neutrophils is that you can treat them in a certain way and break them apart so that you get cytoplasts, that is, a part of the cytoplasm that closes off that's separate from the main cell body, and it doesn't have the nucleus. But these cytoplasts are actually more stable than neutrophils, and they again, don't have genetic material. But they move, in some cases, faster than the original cell. So this could be a really powerful kind of platform to use for this sort of search and delivery function in medicine. We're trying to explore the range of inputs that can be detected, different GPCRs that could be linked to different disease signatures. Can we link different classes of receptors, receptor tyrosine kinases, to this pathway? What payloads and actions can we hook up to this? We're trying to load these things with imaging agents just as a first thing to see if they can find different signals. Could we hook them up to secretion of different products, cytokines that could modulate the immune system, other products? Could they be leaders for regeneration by secreting molecules, for example, that would, like netrin or something, that then talk to neurons or other cells that we want to control movement and differentiation of? Can we hook them up to, can we control things like cell killing? These cells, of course, have the capability of creating a reactive oxygen species. Could we control that as one other kind of action or payload? Ben and a number of other students and postdocs are working hard on these forward efforts. So let me just conclude by saying that in these early studies, we found that it's possible to rewire receptor and GTPase pathways to control a complex behavior like cell movement. What we're really trying to do is expand the functional diversity of these cells by exploiting their native modularity of these cell signaling systems. And I want to make one point here that we've, with this discussion in synthetic biology of parts and kind of designing them, putting them together for applications, to me, I think one of the sort of other things about synthetic biology that really is important to me is its power in allowing us to understand endogenous biological systems and how they're put together, what the points of modularity are, and how nature and evolution generates the amazing diversity of behaviors that we see. We are trying to develop a toolkit of parts and approaches to manipulate cells with high spatial and temporal precision, and I think this is we're really pleased with this sort of start towards engineering cells with at least simple therapeutic functions. So let me end by thanking the people who did the work. The work on the engineering the GEPs was done by Brian Ye in collaboration with Daphne Barsagi, who's at NYU, and her student Rob Bertigliano. The work on the light-controlled GEPs was done by Anselm Levskaya and Chris Voigt, and Ben Rao is the person who did the work on the engineered G-protein coupled receptors. Thank you.
Thank you for a very intriguing presentation. The floor is now open for questions. Yes.
Thank you. Very interesting piece of work on the light-activated motion. Of course, one of the questions is, how do you get the light to where you want it in a system? And I'm intrigued to know whether you've looked at the coupling of the biological systems with non-biological nanosystems to actually target that light in very, very specific patterns and locations, perhaps using technologies such as quantum dots or nanoscale light pipes.
No, that's a very interesting question. I think this notion of cells or particles working together. I alluded to that that these engineered cells could only be a piece of the puzzle. Maybe you'd have multiple ones. And I think that by the same token, bringing in nanoparticles is something that as another part of the system is a great idea. You're talking about, for example, that maybe certain inputs would be more penetrable that then could be transduced into the right wavelengths by those particles. So I think that's a great idea. I mean, I think that is a problem, getting light of these wavelengths into a body. I think this is going to be a really powerful tool right now for cell biology, but you know, we have to think about strategies like that to if it was going to be really applied therapeutically. Yes. No, so were those real time with the light controlled? No, those were sped up, I think, about 100-fold. Ray?
Hi, Ryan. Very nice work. I also have a question about the light-induced interaction. You're depending on some cofactor there. Is that any problem? I mean,
could you say that again?
You're depending on some cofactor there.
Yes, that's a very good point. You guys are all raising the right question. So there is this chromophore. It's heme-induced. So in this case, we're providing that molecule, but it in principle should be relatively easy to, because it's derived from heme, you could introduce this into cells enzymatically. But it is an issue that needs to be dealt with. Other questions?
I just have one question. In terms of the spatial resolution of the light-directed movement, is it right now limited by the biological implementation or by the light source?
Well, you know, we just got a instrument that it's like one of these DLP projectors that can project very complex and specific patterns. So we're really investigating that right now.
Okay, great. Thanks.